He's a tropical biologist. He's studying the ecology of rainforests, both the insects and the plants. So I think this proves to be a very exciting talk that we'll all enjoy. He's the director of the Institute of Entomology at the Czech Academy of Sciences. He's a professor of ecology in the Zoology department at the University of South Bohemia in the Czech Republic. And also since 1977, he's been head of the New Guinea Binatang Research Center in Papua New Guinea. And he became a member of the Learned Society of the Czech Republic in 2009. He's listed on the Stanford list of the 2% most cited scientists in the world. So I'm sure he's going to give us a super plenary session. And I should also mention that his talk has been sponsored by Harper Adams University. So Wojciech, without any further ado, thank you very much, Henry. I look forward to your talk.
Good morning, everybody. Thank you for invitation. Thank you for introduction, which was certainly more informative than my Wikipedia page. And it's a pleasure for me to speak about insects, the little things that run the world, focusing on how they impact big trees of tropical rainforests. So let's start this discussion or this examination with some biodiversity dimensions, asking how many species of trees on one hand and insects on the other hand are there in the tropics. Of course, we entomologists have always known that there are about 50,000 tree species in the tropics. That was because the famous global biodiversity estimate for insects by Terry Irwin included this figure in his calculation, where Terry Irwin studied communities of insects on tropical trees. And then when he wanted to expand his global estimate, then he needed a number of tree species. He didn't know that, he couldn't find it in the literature. So he, being at the Smithsonian Natural History, he asked his colleague next door, Richard Ide, who was a botanist. He also didn't know, because botanists don't count species by life forms, they count that by taxa, by families and orders. And so Richard Ide called his friend on the phone, Richard Howard. He considered the matter very deeply for about 5 minutes, and then he ventured a guess that there might be about 50,000 tree species in the tropics. Terry Irwin included this quite honestly as a personal communication in his paper. And since then, nobody ever questioned that number. Entomologists, including my team, actually questioned a lot of other assumptions by Terry Irwin, but not this one. So it has taken 50 years since a proper botanist, very slick, decided to examine the matter rigorously. As you can see from his PNAS paper, he needed a lot of co-authors on that. And after much effort, extrapolating from lots of plant plots in the tropics, he came, perhaps disappointingly, to the conclusion that 50,000 was right. And so that was his estimate, still wide uncertainty, as you can see. In 2022, there was another study by Roberto Gatti, who came with 73,000 species globally. So that actually increases the number of tree species in the tropics as well, because there is no way how there could be 20,000 extra tropical species. And again, it was done by extrapolation from the plant plots, and that estimate includes 9,000 undiscovered tree species. So now when we go back to the insect estimates, the first known one from the literature is by John Westwood, 1833, where under the impression of all these materials coming from British Empire, he thought that there would be about 500,000 species of insects. John Westwood was, by the way, the first entomology professor at Oxford. He was actually the second professor holding Hope Chair, which was founded by Hope as a benefactor. At one stage in my career, I actually interviewed for Hope Chair, but didn't get it. So From John Westwood, it took 150 years for the more famous estimate by Terry Irwin, which I already mentioned. He studied communities of beetles on tropical trees, then made assumption about host specialization and multiplied by the 50,000 of tree species, and came with a rather surprising 30 million insect species. That generated a lot of research, including ours in Papua New Guinea, where we looked specifically into host specialization and corrected some of his assumptions and came up with 6 million species as a result. Okay, now I did something wrong. Okay, so we came with 6 million species. That was a publication in 2002. So maybe it's time to revisit after more than 20 years what has been the progress in entomology since then. Of course, that was right before large-scale barcoding started to shape the taxonomy of insects. Currently, perhaps the most extensive sampling program which is using that barcoding is Global Malaise program. With a number of study sites. That was also one of the methods which started the whole concern about insects decline. We have run one such malaise trap in New Guinea, where in 1 year there were about 40,000 insect individuals caught by 2 square meters of sampling surface. They were barcoded and they were classified into 5,000 species, which was far from the complete sample. The estimate exceeded 10,000 local species locally. What is important here is that this really expanded the number of species in Diptera and parasitic Hymenoptera. So we started to realize how underestimated the Diptera species diversity was. We can illustrate that on the level of the fauna for some well-known country. Sweden is perhaps one of the best-known countries for insects. Of course, the UK will come very close after that. This is the share of the key orders on the species diversity of Sweden over the years. So in the time of Carl Linnaeus, it was dominated by Coleoptera and Lepidoptera, but their share has been declining ever since. 2003 was the last estimate before barcoding. The estimated one is including the barcodes, and we can see that especially Diptera increased in importance. So nowadays it looks like the Lepidoptera, at least in Sweden, represent only 7.5% of all insect species. Sorry, 4.5%. Coleoptera 7.5%. This is related to the global estimates of insect diversity because Irwin's was based on beetles, assuming quite unrealistically that they represent 40% of all insects. We have corrected that down to 23% in our estimate and 10% for Lepidoptera. If we go to the current estimates for Sweden and correct our estimates, that means we increase the global estimates because the importance of Lepidoptera and Coleoptera is diminished. And then we also multiply it by 1.5 to account for more tree species we have globally. Then we are actually back to double digits in millions of insect species, somewhere around 20 million. So again, this is something to think about. Now, of course, there are more species of both plants and insects in the tropics. We all know that. This is the map of the floristic diversity. But the interesting question is whether these two numbers go together. So if plants are the basis, the resource basis for insects, does the number of insects correspond to the number of plants? Or is there more than or fewer insects in the tropics than expected based on plants? The botanists are surveying plants, of course, by plant plots. This is sort of gold standard, so-called forest geo network, which comprises 50-hectare plots. And currently they are monitoring about 13,000 tree species across the tropics, and more recently also expanding to the temperate zone. I think White Woods is the British plot here. I will be talking about the plot at Barro Colorado in Panama, which was the first one, and then our plot in Vanang in Papua New Guinea. This is how the plot in Papua New Guinea looks like. It's 1 times half kilometer, 50 hectares, with the research station next to it in quite a remote place. And this is the standard procedure, measuring diameter of the stems, painting the point of measurement. So when you come back after 5 years and you measure again, you measure in exactly the same place, tagging each tree, and then the most difficult part, identifying 2 species. This is the result. Every pixel is 1 stem of the tree. So we have 288,000 of individual trees, which we like to revisit every 5 years, how they are doing. And this was the team in the last survey who spent entire year in the tropical forest doing that. So that gives you a scale of these measurements. Of course, as a finance limited scientist, we have to think about costs. We know that, for instance, DNA sequencing followed originally the Moore's law. That means the price was halving every 2 years. Then it really plummeted and got really cheap. When we look at the cost of obtaining these plant data, then I would postulate so-called Hubble's law, which basically says that for the last 100 years, the cost really didn't change for what the botanists are doing. Steve Hubble was, together with Robin Foster, one of the 2 authors of the 50-hectare plot idea, and they got funding from National Science Foundation to do the first one in Panama, 1986. That was actually quite interesting movement in botany standards, because by that time, the gold standard was 1 hectare and diameter of 5 centimeters, which will give you about 1,000 stems per sample. They suddenly proposed 250,000. So if you multiply current best protocol by 250 times and propose it as a new research project, no matter whether it's botany or entomology, you are likely to face some opposition, because people who are doing the business as usual will feel like you are basically implying that they are too lazy or incomplete in sampling. And so it was always a mystery for me how they got the funding for the first project. Interesting book which is called Coexistence, and which is mapping the history of tropical research, especially in Barro Colorado, clarifies that. It was actually an ornithologist on the National Science Foundation panel who liked the project and pushed it through. So that sort of explains it. This New Guinea plot is on the indigenous owned forests in Papua New Guinea, and it sustains 10,000 hectares of protected area. You can see this is the Hansen's online map of the forest loss. And the landowners from Vanang village, which are practicing Sweden agriculture, so you can see some forest loss from their food gardens, are basically protecting 10,000 hectares, unlike all the neighbors who invited logging companies. So this research also provides employment, training, and sustains conservation. Now, as entomologists, we wanted to have the same type of data as botanists. So that means we wanted to have insect communities not tied to some sampling surface of Malay strap, but to a specific area of the forest. So we would like to set up the plot, time the sampling based on seasonality, get all the insects there, get all the plants with the herbivory levels and other species traits. Then, of course, identify everything, barcoding, photography, and so on, and then create the food web interaction between plants and insects, or between different insects for the analysis. So how to do it? Of course, forests are hard to sample for insects. We have used elevated platforms. We have used canopy cranes where available. We also built one in Papua New Guinea. And rather surprisingly, maybe, we also cut down the forest. That's actually a very good way to sample insects from the forest canopy. In the indigenous areas with slash and burn agriculture, that's actually a very efficient way. But we have done that also, for instance, in the US in privately owned forests. So this is an example of extensive sampling in Papua New Guinea, 1 hectare, all the stems 5 centimeter dBH, and their taxonomy on the level of genera. This is the same map with the leaf mining insects for each individual stem. So you can see that there was between 0 and 5 different species, but when we aggregate them across 1 hectare, we get almost 300 different species of leaf miners. This is the food web for the leaf miners and also food web for ants based on ant nests for that 1 hectare. Now, what are we going to do with this data? We want to use this New Guinea tropical sample and to study whether the prediction based on that for temperate zone will be successful for actual empirical diversity of insects there. So we have used the large sample from the tropics and reduced it repeatedly to the level of number of stems, size distribution of stems, and number of species, which we found in smaller plots across the world. And therefore, then looking at what insects these selected stems have, we could predict what is the expected number of insect species based on plant diversity alone. And so these are, in red, there are the plant plots which are used for that prediction across the world. And then the triangles are the plot we actually sampled and verified that prediction. So when we look at these plant plots, of course, there is a latitudinal gradient in species richness of trees, which leads to prediction of similar gradients for ants and leaf miners. Interesting thing is, however, whether plants completely explain that gradient or not. When you look at the predicted and observed number of ant nests per 0.1 hectare, then you can see that for the temperate zone, for the low tree species richness, there is actually a big difference. We have fewer ant species than we would expect from the vegetation alone in temperate zone. The opposite pattern is seen for leaf miners, where the predicted numbers are higher in the tropics than we actually see. So insects respond differently to latitude from the plants, although there are, of course, general trends with tropical maximum. Now, coming back to the vegetation and the effects insects have on its composition. The fact that there are perhaps 70,000, 75,000 tree species globally, most of them in the tropics, is not really very surprising or very puzzling. The evolution can generate a lot of species, as we have seen on the islands. You know, once you have suitable conditions, then species are actually very cheap to generate. Evolutionary speaking. So that's not an interesting puzzle. What is, however, interesting is this huge local diversity. This is a typical 20 × 20 meter square from our Wanang 50 hectare plot. And you can see that there are 220 woody plant individuals from 85 species. And so their spatial distribution and huge local diversity is surprising, because you would expect that every tree, of course, generates a very intense seed rain in the neighborhood of the tree. You would expect seedlings from the same species coming up and then forming clusters of conspecific trees, which you don't see in this pattern. So, of course, naturalists, when they first, with their temperate zone background, when they first came to the tropics, they were really surprised by this huge diversity. This is Alfred Wallace looking at birds of paradise in New Guinea during his travels, but of course the same impression was made on Darwin, on von Humboldt, and pretty much everybody else. So we might say, well, clearly there is something strange about the tropical forests. However, when I bring my Papua New Guinea students to Europe, they are equally surprised by how strange are European forests. You know, where are these hundreds of tree species? You know, they are posting bizarre European forests on Facebook. So the surprise of naturalists is not a good base for ecological hypothesis. So we should ask which of these two types of the forests require ecological explanation. I would still argue that it's tropical forests which are sort of unusual, because normally we would expect, and that's the stuff of all ecological textbooks, that the tree species which happens to match the soil and climatic conditions and is competitively superior will over the generations dominate the forest. That's clearly not happening in tropical forests. So there has to be some kind of penalty which is associated not with a specific tree species, but with the fact that it's becoming more common with abundance. High abundance is disadvantageous, and that will keep every tree under control. That high abundance may be a problem because of pathogens and herbivores. That's the core of the Jensen-Connell hypothesis, independently published by Daniel Jensen, who is an entomologist, and Joseph Connell in 1970s. The idea is explained using insects, and by then Jensen, is that the seed rain is diminishing in intensity with increasing distance from parent tree, but the likelihood of survival is actually increasing because the seeds migrating further, and then seedlings and saplings are getting away from potential source of pathogens and herbivorous insects. And so when you multiply these two curves, then the recruitment is maximum at some distance from the parent tree, which is exactly what we observe in the tropical forests. So when we want to study these in New Guinea, again, we have surveyed number of insect guilds. Each, of course, requires different sampling and rearing techniques. These are just visual example of 12 guilds using same set of 12 tropical trees in the lowland forest in New Guinea. You can see diversity in number of species and their specialization. When you put all this together, we get quite a complex food web. This is only 36 trees. When we extrapolate to 200 trees, a typical local diversity, we will predict about 10,000 insect species feeding on plants in 50,000 individual interactions between plant species and insect species. That's, of course, part of the picture. Then we have parasitoids, another about 13,000 interactions. And so these are very complex food webs. When we studied caterpillars, we found that they have a range of host specialization, but their parasitoids were all specialized. When our colleagues Owen Lewis and his group at Oxford studied leaf miners, they found the opposite pattern, that their parasitoids were quite generalist. So that led us to a maybe suggestion that the completely specialized tritrophic food webs might be forbidden in the tropical forest because you have rare tree species. If you have a specialized herbivore and then you have specialized parasitoid, it might be too difficult for the parasitoid to locate the very rare host in that kind of system. Unfortunately, we went to verify it before we published this nice hypothesis and found that actually it's not true, because when you look at the gallers, then you have extremely specialized system in the tropical forests, and then you also have a very specialized parasitoids. So that's work of the Phil Butterill, and there are 6 insect species at least specifically attached through this food web with each 3 species on average. Now, the rest of my talk will be about experimenting with these food webs, because I believe what Richard Feynman said in different contexts, that if we want to really understand something, we have to either be able to create it or at least predict what will happen if you somehow disturb it. And so that's the benchmark we want to achieve for food webs in tropical forests. So the Jensen-Connell hypothesis can be tested in different ways. You can look at spatial variation of the vegetation, looking at different ages, seedlings, saplings, mature trees, or you can revisit in time, like we are doing in Forest Geo Network. And that could be either natural vegetation or specially planted one. There is actually a proliferation of tropical experiments, planting tropical trees in different combination of species richness. Or, and that's our main focus, you can manipulate these enemies, including insect herbivores, on the native or planted vegetation. So that's something I want to show here. Historically, first prominent such experiment was by Owen Lewis and his PhD student Richard Becci. They had these 1 meter square plots with seedlings in primary tropical forest, and they were spraying them by insecticides and fungicides, looking at what will happen. This is basically an experiment focusing on Jensen-Connell hypothesis, and it has been 40 years since this has been first attempted, which is, quite a long time, not very good sign of experimental mindset in ecology. And they found that one type of fungicide, when you apply it, then the diversity of seedlings declines. And that's exactly what you would expect with Jensen-Connell. If you have a certain species, this is example of one of the trees there in the neotropics, at different densities, then the seeds will recruit different sapling density. Of course, the more seeds, the higher number of saplings. But when you spray the fungicides, then you will see that suddenly, when released from fungi, the common species or high densities of seeds are doing better than they were doing previously. So that's the negative density effects which supposedly fungi should have. All these are black box experiments. Basically, we don't know what we are spraying in terms of fungi or in terms of insects. So that's one of the problems. I will return to that later, but I would like to mention that actually the unwitting experimental test of Jensen-Connell had been done 50 years before Jensen-Connell published their hypothesis, and that was in the brand new city called Fordlandia, which was started by the Ford company in the Brazilian Amazon in 1928. They wanted to have steady supply of rubber for their booming automotive industry, and so they set up a sort of American type city in the jungle and then planted huge amounts of rubber trees around it. Unfortunately, a rubber tree is a tree from the Amazon. And so this was a high density experiment, and it predictably led to the fact that the rubber trees all died because of fungus. Apparently, the experiment also didn't go well socially. There are records that the native workers grew tired of the American food and revolted in the town's cafeteria, chased away the managers and the town's cook into the jungle until the Brazilian army arrived. So apparently there is still ruins of Fordlandia, which you can revisit. Now, there are case stories which confirm Jensen-Connell when it comes to insects and their predation of seeds. This is one of such nice stories, Conotrachelus beetles attacking seeds of Virula surinamensis in the neotropics, perfectly works as predicted by Jensen. The problem is that this is a proof of the concept, but whether this effect is widespread in the communities enough or not, that's another matter. My colleague Yves Basset led a large-scale survey of seed predators in tropical forests in 3 places, Panama, Thailand, and New Guinea. And he found a very large local differences, already starting with the host specialization, which was much higher in Panama. The host specificity is prerequisite for Jensen-Connell to work. You have to be focused on a single tree or a few related trees for that. Our surveys in New Guinea looked into more than 300 tree species and their seed predators, and we found that although there is a significant fauna, lots of these trees, majority of these tree species do not have sufficient rate of seed predation to be consistent with Jensen-Connell hypothesis. Of course, that doesn't mean that these seeds are not eaten in large degree by insects. It only depends on the Jensen-Connell effect, depends whether there is a relationship with the density of the seeds. This was another experiment where the artificial communities of seeds were assembled on the forest floor, and then some of them were sprayed by insecticides. You can see that there was again a variety of responses. Some species were not affected. That means there was no insect impact on the seeds or seedlings. Some of them were benefiting from insecticide already from the seed stage and then continued in the seedlings. Some of them, seeds were not affected, but the seedlings were, and so the advantage of the insecticide plots was growing over time. So you have all kinds of responses, but the experiment which Owen Lewis did with Rob Bekci is the one which is proving whether there is a Jensen-Connell effect. So we have done that in New Guinea. Lucy Houtkova, as a PhD student, used again 1 meter square seedling plots in the 50-hectare plot. So there were 200 clusters, two were left in each cluster, two plots were left as control, one sprayed by fungicide, one by insecticide. What happened is that what sometimes happens is that your experiment doesn't go as planned. Here, fungicides didn't have any effect. Insecticides did, but they have opposite effect. When you spray by insecticide, then the number of seedlings goes up, not down. That's because the number of individuals goes up. So basically, in this particular case, if you have a hypothetical community of seedlings, every color is different species, then Jensen-Connell effect should work like this. Insects attack preferentially common species, seedlings die, new seedlings recruit, some of them from new species. Species diversity goes up. What really happened is that, yes, insects attack seedlings, indiscriminate of their density, they died, no new grew, so therefore some species were lost. However, when we looked at seedling survival, the picture is different. So I think the question is still open, because when you look at the seedling survival, it was negatively influenced by the abundance of conspecific trees around the plot. So if you really have a tree from the same species, it's bad for you as a seedling. But then there was a positive significant effect of interaction between insecticide spraying and conspecific trees. So that negative effect of the tree disappears when you remove insects. So that's a very strong indirect evidence that there is some kind of impact of insects coming from parent tree down to the seedlings and killing them. Now, not all insects are enemies of trees. We know a lot of mutualistic relationships. The most famous are ant plants. We have Cecropia in the neotropics. We have Macaranga in Southeast Asia, Endospermum in New Guinea. And so if they are working towards helping survival close to the parent tree, that we can see as a sort of reverse Jensen-Connell effect. And so instead of recruitment of seedlings at some distance from the tree, you would get maximum recruitment close to the tree. And then you will form conspecific clusters of trees. So we tested it in our 50-hectare plot in Papua New Guinea. This psychedelic picture, it's all the ant species and tree species in the plot. So it could be significant. And this is example of a common species, Schizocheton laciocarpus, with hosting two ants, Podomyrma and Anonychomyrma, with Shafia Zahra and her supervisor Tom Fail. When they prevented access of ants to the seedlings close to the parent tree, then indeed the survival of seedlings went down. So especially with Podomyrma, the blue line, instead of 95% survival after 1.5 years, you had only 65%. So it looks like, yes, ants can act as a reverse Jensen-Connell agents. The most striking example perhaps is so-called Devil's Gardens in the Amazon, where the local people sometimes came across a perfect monospecific garden of Duroia hirsuta, which is an ant plant, and somebody planted it there. One of the postdocs, Megan Fredrickson at Harvard, found out the mechanism. The ants from Duroia hirsuta actively kill any seedling and sapling which is not Duroia hirsuta, and by that way basically open a possibility for monospecific growth which is expanding. Some of these Devil's Gardens are estimated to be 800 years old. Fortunately, when the garden gets too big, then the control of ants disintegrates. Otherwise, you could actually think about extreme where over the thousands of years, entire Amazon would be converted into a huge plantation of Duroia hirsuta. Another aspect I want to consider is what is the importance of insects in rainforest regeneration and early succession, because that's increasingly important section of the tropical forests. And it's quite different from primary forests. The plants there, the pioneer plants, are strongly selected for large, fast-growing leaves, which are built very very economically, so they have small leaf mass per area, therefore they have a short lifespan, but at the same time they have a large photosynthetic capacity, they are growing fast, therefore they have a lot of nitrogen and phosphorus. So these are basically pioneer species maximizing the growth and reproduction at the expense of lifespan and expense of defense against insects. Why is that? Because their main competitors are other plants trying to overshadow each other rather than insects. And so whether insects will be important, you can argue both ways. You can argue that because this is mainly about plant competition, the plants with the fast growth can easily compensate any herbivore damage, and therefore insects will not be important. You can also argue that because it's really very tight, very intense competition, any damage by insects can tip the balance between competing plants. And so we try to find out. Regenerating forests are increasingly important because of the anthropic disturbance. This is shown in recent review by Robin Chasdon, but also her earlier book, Second Growth. But the secondary vegetation is important also naturally, not only in tree gaps, but also in many areas, including New Guinea. You have a very intense dynamics of landslides, often triggered by earthquakes. This was already pointed out in a Nature, in a science paper in 1979. And this is example 6 years old from the intact landscape in highlands of New Guinea after a major earthquake. You see all these natural landslides which open forest to regeneration. Even in the 50-hectare plot we have there, we see 2 or 3 smaller landslides every year. So secondary vegetation is an important part of the forest dynamics. And so we can study the importance of insects again by manipulation of natural vegetation. In New Guinea, where people are practicing slash and burn agriculture, you can experiment with rainforest as much as you like without causing any extra deforestation. People will be actually much happier if you pay them for looking at insects than buying sweet potatoes they grow in their gardens instead. And so this is example of such experimental design when there are secondary artificial tree gaps basically in the place of the future food gardens with replicated plots, some of them again sprayed by insecticides, fungicides, some of them preventing bird access as insect predators. That's work by my PhD student Piotr Schäfer. In lowlands, you can see after 1 year you have nice 4 meters high spontaneous pioneer vegetation there with lots of species. And we found that, yes, here insects actually do keep even secondary succession diverse, and they do change plant composition, and they also make the succession more predictable. In these replicated plots, every succession starts based on what seeds are in the seed bank. But then the effect of insect basically streamlines that successional trajectory into the same track. We also can look at enemy manipulation in a manipulated vegetation. That's a work of my current PhD student Kari Yamba, again in New Guinea, where again this kind of overgrown garden has been nicely converted in experimental plot. And we have control insecticide treatment, and then we remove repeatedly all the herbaceous vegetation because that's known to slow down succession towards tropical forest. And then the combination of herbaceous removal and insecticide. So this is how it looks like, different plots. And you can see that the removal of herbaceous plants, as expected, has a visible effect on promoting woody plants. But it's more complicated. Individual species either benefit from this kind of disturbance or are indifferent to it or are promoted, even among the herbaceous plants which are being removed. After the end of experiment, we harvest all the vegetation plus insects on that vegetation, and also look at the effect of elevation here. Again, the impact of insects with elevation, we can argue both ways. We can say that in the tropics, with every 1000 meters elevation, you get 5.4 degrees colder. So of course, that's not good for insects. So you can expect that high elevation means a lower impact of insects. But also you can argue that in the lowlands there is an intense predation of insects, birds, bats, ants. They all have maximum in the lowlands, and so when you release insects from that predation pressure, then maybe the impact on vegetation will be higher. Okay, I have done something again. No, okay, it's working. So the results, when we removed insects, then as you can expect, the plant biomass increased in both non-woody plants, woody plants, and therefore total vegetation, but only at low elevation, 700 meters. There was no effect at 1700. So it looks really like insects impact is going down with elevation. The same goes for plant diversity. There again, insects were keeping the vegetation, especially woody plants, more diverse. When we removed them, then diversity went down, but again, only at low elevation. The removal of non-woody plants then increased the woody plant biomass, and then also increased, decreased the herbaceous biomass, increased the woody plant biomass. These two contradictory effects perfectly compensated each other. So the total vegetation biomass was without change, whether we removed all the herbaceous plants or not. That would probably change over the further succession. This experiment was 1.5 years long. When we combined this removal of non-woody plants with insect spraying, there was no additional effect of insects. You can see that basically the results are always the same. The same goes for the species composition of woody plants. In the low elevation, insects were those which were driving the change compared to control, not the removal of herbaceous plants. In the high elevation, the opposite pattern. Insects had no effect, but removal of herbaceous plants did change the composition of woody plants. Now, we hear a lot about climate change. What is the effects of that in the tropics? Well, it's a textbook knowledge that for the early successional vegetation, the source of that vegetation is in the seed bank. And when you take soil from primary forest and germinate the seeds there, you get the same array of pioneer plants as you get from the secondary forest. That's what we confirmed in this experiment at 1700 meters in New Guinea. And then from the primary forest, for the woody plant, we germinated from the seed bank mostly pioneer species, which you never see in the intact primary forest, so they are just waiting in the soil. There was one alien pioneer species, Piper aduncum, and there were a few primary forest species also germinating. The herbaceous species, half of them were alien. Yeah, so it was perfectly intact primary forest in one of the most remote places you can imagine. That's the Sarawak Mountains, where we have to fly by small airplane, land on the airstrip, hike 3 hours over the range to the place where there have never been in recent years any human habitation, go to perfectly wild primary forest, take soil from there, germinate it, and half of the herbaceous species are alien plants. So that gives you an idea, how basically how the alien plants are overriding the entire planet. So this is our experimental situation. We thought, well, if the pioneer community of plants is almost entire in the seed bank, if we move the soil with the seed bank, that's a perfect experiment simulating climate change, because we have these very predictable decrease in temperature with elevation. So at 1800 meters in our field camp, we moved some soil locally, and then we also brought the soil for the spontaneous succession from 22 and 2400 meters, which simulated global warming 2.7 and 4.1 degrees. So quite strong one. We have constructed little cable cars, transporting the soil down. We are talking about tons of soil through very, very difficult terrain. Also, hand carrying the soil with the local assistants, mixing it to homogenize our replicated experiment, spreading it in the experiment. This is still ongoing, so I can't tell you what is the insect effect, because we will again exclude insects or not. But when we can already look at some early succession. These are the control plots at different elevation. And when we brought the soil to 1800 meters, it looks exactly the same, no matter whether it's 2400, 2200 or 1800 meter soil. It's all dominated by alien herbaceous plant Crassocephalum crepidoides from Africa. At 2400, there is still not much growing, but interesting is 2200, where there is a completely different community dominated by a different plant, Heliotropium longifolium. So we are wondering with climate change whether insect and plant species will be climbing up the hills. We already know that in Europe it's happening already with insects, that they are expanding the ranges up the mountains. In this particular case, it has, for plants, it has already, for pioneer plants, it has already happened. They already dispersed, and they are just waiting in the soil for the climate change, and then they will start growing, as you can clearly see here. So to conclude, for the Jensen-Connell impacts of insects, insects are important predators of seeds, but may not always act as Jensen-Connell agents on the community level. Insects can be Jensen-Connell agents promoting the diversity of seedlings. They can be also reverse Jensen-Connell agents, decreasing diversity of seedlings. They are important in maintaining plant diversity even in pioneer succession, but their impact decreases with increasing elevation in the tropics. So that's more or less biological content I have to offer here. I just note at the end one issue here. When you look at the tropical forests, then the typically 1% of species represents 20-25% of biomass. And that's because of Jensen-Connell, keeping really greedy species under control. Okay, in human society, the top 1% of people own about 50% of wealth. So the question is, should we consider Jensen-Connell mechanisms to alleviate that? Actually, when you think of money, money behaves as positively density-dependent agent. Basically, large sums of money grow faster rate than small sums of money. And so we need to consider some kind of opposite effect. You can say in biological terms that progressive taxation is a density-dependent regulation of money growth. And so, we can argue for that. There are, of course, other proposed solutions, but that would be probably for a different lecture. So finally, I would like to say that, you have seen most of examples from Papua New Guinea. It's fantastic country for the fieldwork. There is a lot of experiences. This is Mount Wilhelm, the highest peak in the country, where just one story from the field. We were facing the need to set up for one particular project 8 camps at different elevations at the same time, and with the help of local people, do a lot of insect surveys there. We were concerned that if our staff started paying them, then we would be vulnerable to attack on these camps, because if people know there is a lot of money. So we decided that we will issue these kind of receipts, and then I promised everybody that after 2 months, when the project is over, I will come with armed police, with big bag of change, and whoever presents receipts will be paid the nominal value on the receipts. That indeed happened. Everything was a success. But I was expecting a long line of people at every elevation clutching few receipts as they worked. That did happen, but also at every elevation, there were few people who presented hundreds of receipts, worth of thousands of pounds. And they were legitimate, so we paid them, but I was wondering what was happening. And then I found that we actually unwittingly started a new currency on the slopes of Mount Wilhelm, because people there who wanted cash quickly, they wanted to buy something in the local village store, they used these receipts. And they were accepted by local traders, but at less than face value, so they also made profit. There were even families with relatives in town which were paid, and so they have cash. And so immediately there were a class of bankers which emerged, and they started actively buying these receipts at even 70% of face value. And so during this experiment of sort of introduced capitalism, about 50% of all receipts concentrated in the hands of these few people and each elevation. So I would like to thank our staff in New Guinea Binatang Research Center, my colleagues in České Budějovice and the Czech Republic, our funders, and finally, I would like a few mention about the country of Papua New Guinea, which by coincidence is celebrating 50 years of independence next week. It's a post-colonial country in the eastern part of the island of New Guinea. The British actually did not want to colonize Papua New Guinea. They only did it very reluctantly to stop the Germans again. And so I would say island of New Guinea is the third largest tropical rainforest. New Guinea is also the world's richest island flora. That's a paper in Nature in 2020. It's historically also, that was the last big contact between civilizations. In 1930s, there were 1 million Papua New Guineans living basically in the Stone Age technology. Nobody knew about them. Australian patrols got into contact. And it's actually very interesting and very happy history. It was one of the few contacts between vastly different civilizations which went on a fairly agreeable terms. So, you know, it's very sort of heartening to read about it. New Guinea, I didn't touch on that, is insanely culturally diverse. It has 15% of all the languages in the world. This is lay secondary school where the students there are speaking 126 languages. So, you know, it's completely incredible. It's also one of the few post-colonial countries where democracy sort of works. This is the father of the country, Michael Somale, on the banknotes, who was voted out of office after the first election, spent time in opposition, then was voted again to the government, then was voted again to opposition. Exactly how it should happen, but it does not in you know, many post-colonial countries. And finally, this is the German-based Academic Freedom Index evaluation, and New Guinea is actually doing better than Great Britain, as you can see. So this is all from me. Thank you. Happy to answer any questions.
Wow. Well, thank you very much. What a brilliant talk. I really enjoyed that. Do we have any questions? We're officially out of time, but I think we need to at least 5 minutes. I'm not sure whether the organisers agree that we do 5 minutes for questions. Okay, we will. Has anybody got anything they would like to ask Vojtěch? Graham.
So, amazing diversity of plants and herbivores and enemies and everything. How predictable are relationships if all you know is the phylogeny of the plants and the herbivores and the insects? Are there strong patterns of relatedness through there, or is it all driven by other traits?
Yes, I would later on you can catch up with Simon Seeger, who is here in the audience, and he actually did exactly this kind of study. If I remember, it's a significant, the predictive power of phylogeny are significant. This was studied on caterpillars and trees. And so if I remember, about 50% of variability can be captured by phylogeny. This is one aspect which I completely ignored here, but it is another important sort of element of these food webs, the phylogenetic predictability.
Hi, thanks for a really interesting talk. I don't know if you've come across the work of Rudolf Meyer and the dark taxa, with the multiple malaise traps around the world. And he suggested that for insects, at least in some cases, there's the reverse of the latitudinal relationship between species richness and latitude. I wondered, do you have any evidence for that in the in Papua New Guinea?
No, I have to say that using Sweden as a as an example to be applicable to tropical areas is a bit of a stretch, and so I would say we don't know. There is this discussion about the reverse gradient for ichneummonid parasitoids, for instance. We actually looked into that, and we sort of did not come with a strong evidence for that or against that. It's quite hard. There are some examples which are well established, like aphids, which really are missing in the tropics, more or less. But overall, I think the jury is still out on that.
Great talk. Thanks very much. You've talked a lot about specialization. What about generalization? What about polyphagy? Is that widespread or really rare?
Yes, that's something I would like to focus more on, because there is a minority of generalists which can be extremely polyphagous. We know, for instance, one of the experiments I didn't really describe in much detail was in addition to insecticide and fungicide spraying, we were also adding an extremely generalist flightless curculionid to the vegetation to see what they will do. My experience is that these extremely generalist species are always minor components of insect communities. They are never dominating. So the dominants are typically specialists. And so that would be very interesting to look into what is keeping these generalists down, because clearly the food base, it's not. And so that's kind of a mystery. Generalists do exist there, but they are sort of kept down by specialists. Why? Hard to say.
Okay, I think we probably should stop there. It's now 10:20, but thank you again. That was a fascinating talk.